Hello everyone, welcome you all in this video lecture. So in this video tutorial,
let's start with the introduction
about genome
before
editing.
moving to the topic, let's understand that what is the genome. So the term genome is used for all the genetic material of an organism, and we know that there are nucleic acids named as DNA or RNA which act as genetic material or hereditary material for all the cellular organisms. In the case of several RNA viruses, the RNA molecule acts as a hereditary material, but most of the cellular organisms consist DNA as the genetic material. So the genome is consists of DNA and sometimes as RNA, and it have both genes and non-coding DNA sequences as well as mitochondrial DNA and chloroplast DNA. Basically, the genome of a particular organism consists all the genetic material of the cell or of the organism. And we know that there are two cellular organelles in the particular organism named as mitochondria, and in the case of plants, the chloroplast. And these both cellular organelles contain their own genetic material. So in the genome of the organism, all the genetic material from nuclear DNA to the mitochondrial DNA and all the genes which can be coding as well as non-coding DNA sequences consist. Now we know that the genome or the DNA is made up of genes, and these genes are the unit of hereditary information in all cellular organisms. They are discovered by Johansen, and he also termed them as a unit for hereditary information. And genes are the factors which are responsible for protein synthesis and translation. Now the study of gene is called as genomics, and whole genetic material consisting of genes of a particular organism is known as genome. So let's understand about the history and discovery of the genome editing. So basically the concept of genome editing was started by the discovery of nucleic acids by Friedrich Misser. So Friedrich Misser discovered DNA and RNA, and later on Watson and Crick found the double helical structure and the chemical structure of DNA molecule. So they are also awarded by Nobel Prize for their discovery. Later on, a scientist named as Frederick Sanger and many other scientists from worldwide were independently able to invent a method of genome sequencing. So after that, scientists were able to map the human genome as well as genome of all the cellular organisms. So with the invention of human genome, or with the sequencing of whole genome of the human and other cellular organisms, the genome editing began, and it converted to the reality with the discovery of nucleases and different other epigenetic editing tools. So with the discovery of these genetic tools, the genome editing concept turned into the reality. Now let's understand about the process of genome editing. So basically the genome editing is the technology or a technique of genome engineering which involve different types of processes, and it is a process of editing in a particular genome of a particular organism. So it have different methods. The most common method is double strand break repair, which is the repair mechanism in DNA molecule, and it is used as a process for genome editing. So basically the scientists use different enzymes to cut DNA, creating a double strand break, and they use several types of restriction endonucleases or restriction enzymes which are very specific for the sites, and these have the ability to cut the DNA molecule, and that's why they are also called as molecular scissors. After that, there are two major pathways for double strand break repair. So these breaks are repaired by two major pathways. The first one is non-homologous end joining, and the another one is named as homologous direct repair. Both the processes or both the pathways are repair mechanisms which are found in DNA molecules. So in the case of non-homologous joining, the pathway repairs double strand breaks in DNA without any template, and it is called as knockout mechanism. And in the case of homologous direct repair, it requires a template to join the of the breaks of DNA molecule. So these are the two major pathways for the process of genome editing. Now the process of gene editing requires different techniques for the completion of genome editing, and there are several major techniques which include restriction enzymes, zinc finger nucleases, TALENs gene editing and CRISPR-Cas9 gene editing. So basically the first technique is use of restriction enzymes. So the restriction enzymes are the class of enzymes which are also known as restriction endonucleases and molecular scissors. So as their name indicates, they have the ability to cut down the nucleotide sequences of DNA molecule, and they are very specific. That's why they are called as restriction enzymes. The another technique is zinc finger nucleases. So the zinc finger nucleases are composed of two parts, which are an engineered nuclease which is fused to zinc finger DNA binding domains, and these domains recognize a 3 base pair site on DNA which can be combined to recognize longer sequences. So thus the zinc finger nuclease increase the recognition potential. The another method is TALENs gene editing. The TALENs were named for transcription activator like effector nucleases, which are called as TALENs commonly. So they are structurally similar to zinc finger nucleases because both the methods use nucleases to cut down DNA sequences, and they use dimerization for their functioning. But the DNA binding domains differ in both of the techniques. The fourth technique or the major technique which is evolved recently and used for gene editing is called as CRISPR-Cas9 gene editing. So the CRISPR-Cas9 is a method named for CRISPR associated proteins which are used for gene editing. This is discovered by Jennifer Doudna and Emmanuel Carpenter, and both the scientists were awarded by Nobel Prize for their discovery. CRISPR as a gene editing tool has get popularity nowadays. So now let's discuss about applications of gene editing, or how the process have significance, or how this process is useful for human beings. So the gene editing could be used to modify the disease causing genes in embryo. So there are several genetic disorders like diabetes, thalassemia and many other genetic diseases which are inherited to the offsprings by their parents, and these are some diseases which are caused due to point mutations and several mutations in the genes of the particular organism. So with the help of gene editing, the disease causing genes can be modified or they can be replaced by the proper genes. And the genome editing is used in different gene therapies. So basically the gene therapy is also a medical therapy which is widely used for the cure of different diseases like cancer and many other diseases which cannot be cured by several conventional methods. The gene editing also used to create chromosomal rearrangement with the help of editing in the genome of the organism, and it is very useful application for the method of transgenic animals, also for the formation of transgenic plants. So with the help of transgenic and transgenic plants, we know that both the animals and plants are very useful and have a great economic importance for human beings. So for the formation of these transgenic animals and plants, the method of gene editing could be used. And it is also used for endogenous gene labeling. So instead all the applications or all the significances of genome editing, there are several disadvantages or limitations for this genome editing process. The major disadvantages or the major limitation is the ethical concerns with the genome editing. So there are several questions like, is it okay to use gene therapy on an embryo, and what if the gene therapies are very much expensive so that anyone cannot access and afford them. The another limitation are the safety concerns with the methods of gene therapy, because the process of gene therapy use viral vectors for their process, and these viral vectors can affect or can infect the cells of the body, which causes several infections also. And the genome editing also could make irreversible changes to the every cell in the body, which can affect negatively the organism. And there is a ethical concern with this process that it can become a tool for selection of desirable traits in future children. So with the help of genome editing, the person can choose its desirable traits like intelligence, attractiveness, and many other traits in its future children, which can affect the ecological balance, and it will create the ecological imbalance. Now the process of gene therapy use viral vectors which can affect more cells of the body, and it might cause tumors also in the body, which can be malignant or which can cause into